Welcome to this dialogue of the European Heart Journal. I'm Carlo Patrono, Associate Editor responsible for the weekly journal scan section of the journal. Co-hosting this dialogue with Professor Baigent are the key members of the weekly journal scan team. Associate Editor Giovanna Liuzzo, who is Professor of Cardiology at the Catholic University School of Medicine in Rome, and Associate Editor Massimo Volpe, who is Professor and Chair of Cardiology at the Sapienza University of Rome. Today we have the privilege and pleasure of talking with and listening to one of the leaders in cardiovascular medicine, Colin Baigent, who is Professor of Epidemiology at the University of Oxford. Professor Baigent has been involved in the design and implementation of some of the most important trials in the cardiovascular field, assessing novel treatments for cardioprotection as well as renal protection. Also, he has coordinated several treatment trialists collaborations aimed at summarizing the evidence on the efficacy and/or safety of pharmacological interventions of cardiovascular interest, including antiplatelet, lipid lowering, blood pressure lowering, and non-steroidal anti-inflammatory drugs, just to name a few. Currently is chairing the European Society of Cardiology Clinical Practice Guidelines Committee. So Colin, thank you very much for accepting our invitation. And would you like to share with us and our readers your personal experience in linking these fundamental elements of evidence-based medicine?
Sure. Thank you very much for asking me to talk and to have this discussion with you today. I started off originally reading mathematics, and when I came to medicine as a second degree, I was very interested in epidemiology, naturally, interested in the numbers around medicine. And right from the very beginning, my interest was really to try to make sure that any research I did was reliable, and that if you get the numbers right and you can work out what interventions are effective and you can apply that learning to big populations, then you can hope to make a real difference to human suffering. So that was my kind of grand philosophy when I started out. And it quickly became apparent to me when I had the privilege of joining the team in Oxford led by Richard Peto and Rory Collins. I was very lucky having that opportunity early in my career. I realized that my focus was going to be on randomized interventions, randomized trials, and then meta-analysis of those randomized trials. And it's only latterly that I've become interested in linking in with guidelines, which obviously help to assemble the evidence in a somewhat more gestalt manner, and as a result, has an impact on how doctors actually use treatments or interventions or interpret evidence. So that's really been the background to my career, the backstory.
Thank you. Giovanna, would you like to take over?
Thank you very much, Carlo. This is a great opportunity for me, I think, for our readers. I have so many questions for Professor Baigent, but let me just ask two things that I really would like to ask. The first, during the COVID-19 pandemic, so-called platform trials such as Recovery, but also Solidarity, have played a very important role in providing a rapid answer to clinically relevant questions on the efficacy and safety of several proposed drugs for the treatment of the disease and the prevention of its serious complications. Would you like to comment on the pros and cons of the adaptive design and other features of these trials for us?
Sure. Well, I should say straight away that I'm not personally involved in Recovery, although it is being coordinated by Martin Landray in my own unit and Peter Horby in Oxford. But I am aware of how they prepared to launch the trial and how it's sort of evolved since then. And I think the key thing that we needed with this trial was for things to be quick. Most of the time, if you're preparing to run a trial, you can think about a timeline of months or even years before you can randomize a patient. And that wouldn't have been any good for us. We had to randomize patients whilst the epidemic was in full swing. And of course, what we now know is that it goes in waves, and we've been able to sustain recovery over a period of more than a year, each time picking up on the waves as they come through. And in order to make it quick, the protocol had to be very simple. The processes, the methods had to be very simple. So simple web-based forms to enter patients with a very minimal data collection. Simple methods for recording outcomes and simple outcomes, so mortality, ventilation, very simple. Because what we really needed to know was, among the various treatments that were being considered in this platform trial, which of them had any impact on mortality or on the need for ITU care and ventilation. And recovery has been spectacularly successful in doing that. First of all, coming forward with dexamethasone, and then subsequently, more recently, the Regeneron antibodies have been shown to be effective, and so on. But this wouldn't have been possible if it hadn't been for a number of organizations coming together and saying, right, we've got to do this very rapidly. So Martin and Peter quickly wrote the protocol, and they had contacts within senior levels within the health organizations within UK, NHS and Department of Health. And it was possible to get everything approved within a matter of days. The MHRA approved everything quickly. And so the first patient was entered really within weeks to the trial. And this is quite unprecedented, I think, anywhere in the world. But it meant that they were able to randomize patients right from the very start of the epidemic. And what we now have is over 40,000 patients randomized over a period of a year. But in the first wave, which was the one we all remember as being dreadful, we had patients being entered very rapidly and quick answers to the dexamethasone question within the first wave. So I think the lesson is that we've got to go back in time when it was possible to do this sort of thing more routinely. If you remember back, the cardiologists remember the ISIS trials and the GC trial, the idea that you just have simple data collection and get the answers that you want. Of course, there are downsides to doing simple trials, to doing, I prefer to call them simple or streamlined trials rather than pragmatic trials. But simple trials, they tell you something important about the management of a patient, but they will not tell you what happens to biosamples. They won't tell you something detailed about the mechanism of disease. In general, they won't do that, and they're not designed to do that. They're designed to answer a question that requires a simple design rather than something complex. Those types of studies have their place, but what we needed with the recovery was something very rapid and decisive, and that's what we got.
Thank you very much for sharing with us these important insights. Now I have another question for you. Last month, you have authored an editorial in the New England Journal of Medicine about the Adaptable trial. And you have praised the range of innovative and low-cost methods adopted by the Adaptable investigators to simplify the identification, recruitment, and follow-up of patients. If you'd like to comment on the main features of so-called pragmatic trials that may reduce costs and enhance feasibility of conducting large trials addressing important questions, are there any drawbacks?
Well, I think that, I've never been a great fan of the term pragmatic trials because it sort of conjures up this image that that you try to take a study question and embed it within the sort of mess of clinical practice. Everything is quite messy in routine care. And it's sort of the idea that it's closer to the real world is really very far from the truth. In truth, if you're running trials, doing something in a streamlined, simple way is actually quite a lot harder than doing it in a complicated way, because you actually have to think about what's really important. And you have to sort of engineer your methods so that they really are neat and they give you something that is what you want, but without the surrounding mess that is we see in clinical practice. So I think that the effort that was made by PeopleNet, the organization in the US that put together the adaptable trial, was a really big step forward in trying to move towards, remember in the US, typically trials will cost hundreds of millions, if not billions of dollars. And they've rejected that, and they've come up with this way of, and the key is really to find ways of connecting with patients that do not involve face-to-face contact. First of all, you find patients by interrogating databases of routine healthcare data, so electronic health records. In the US, the way that's done is by taking at each center, they had about 40 centers, each center was responsible for setting up a query within their databases and coming up with suitably qualified patients to go into this study. The study, of course, many viewers will know, is a comparison of high dose of 325 milligrams of aspirin versus 81 of aspirin. And the idea was that it may, in clinical practice, there felt to be uncertainty about whether 325 might be slightly better, might be slightly more effective than 81. So they set this trial up of 15,000 patients recruited from 40 centers by using the databases to identify the patients and then invite them to log on to a portal where they could be randomized and then followed up subsequently every 6 months or 3 months. And the principles of the trial are exactly similar to those that have been used in our own part of the world, in Oxford and in Scandinavia, and there are some other places where if you have electronic health records, you can do this type of trial at low cost. And my editorial was really to point out that this method will lead, if it's utilized, to many more opportunities to answer important health questions. I did mention that I felt that some of the problems that were experienced during the trial could be obviated by having a pilot study. I think this is something that is not used often enough when trying to do big trials. The pilot study is an excellent opportunity to get it all wrong, because when you get it all wrong in a pilot study, you can twiddle around with the methods, get them right before you get started into the main event, the main recruitment drive. You do not want to have problems with your trial when you're trying to recruit thousands of patients every month. So the pilot study would have helped, I think, but it was not detract from the fact that this is a method that has viability for helping doctors and their patients to learn more about the treatments that are really effective. And I do hope that the sort of slightly negative tone of my editorial is not overinterpreted. This is a really big step forward, because if it can be done in this way for many more trials, then we will have a real step forward, I think.
Thank you so much. It's a great pleasure for me to be part of this.
Thank you, Colin. As you know, another example of a large, simple, randomized trial is provided by ASCEND, a study of cardiovascular events in diabetes. which was designed and coordinated by the Nuffield Department of Population Health at the University of Oxford, where you are director of the Medical Research Council Population Health Research Unit. Now, can you comment on the main differences in study question and design between adaptable and ascend?
Sure. Well, adaptable was comparing 2 doses of aspirin in people with established atherosclerotic vascular disease. A wide range of different types of manifestations of disease were seen in that trial. Whereas in ascend, the question was instead whether aspirin is effective, low-dose aspirin is effective for the primary prevention of serious vascular events, heart attacks, strokes, and vascular death in people with diabetes, but who have not yet had any kind of history of vascular disease, not known to have vascular disease. And this has been a question that has been very topical for some time. The question is, well, if you go back now and think how the trial evolved, it was designed to answer that question, as well as the other question in a factorial design of the question of whether fish oil is effective for the prevention of serious vascular events. So, in fact, there were 2 questions in one trial. Another thing that actually would benefit from being used more often as a strategy in clinical medicine. But the way it was done was really novel, I think, in the sense that, well, at least for the UK, it was used for using a mail-based approach. So, and the idea is a bit like marketing. You know, if you want to reach people, then you have to have millions of leaflets printed and sent out. And if you do that, then if 4% of them end up buying your products, then you'll achieve what you want to do. And this, I use the figure 4%, that was the figure. We were able to identify registries of people who are diabetic and followed up for eye disease in various parts of the UK. And by assembling all those registries together, we were able to identify around 450,000 people in total who were mailed an invitation to enter into the study, and ended up out of all that, after they'd then responded to the invitation, potentially been invited to enter a run-in period, placebo run-in period, and then been randomized, we whittled that down to 15,000 randomized. So if you're thinking about designing a trial like that, you've got to have access to the 450,000 in the first place. And that's the key. To do big trials, you need to have very large numbers of people that you can invite. And if you think about it, the idea is that you don't wait for people to come through the door into your clinic and then invite them. That takes forever to do. What you have is a very big pool of people, the 450,000, that you can draw from straight away to identify your sample. So mail-based approach to that allowed us to randomize patients, 15,000 patients, and then follow them up. And as many of you will know, that resulted in a marginally significant, but still a result on aspirin that was consistent with the other evidence for primary prevention of aspirin in people without diabetes. So it was a success in that sense. But it did take 6 years in total to assemble enough patients to invite. So there's plenty of scope for improving the methods. And in fact, that's what we've been carrying on with doing in Oxford. There's an ongoing interest among those who are running the trials in improving the way we identify large numbers of patients and invite them into trials to make trials more efficient. And so more recently, for example, in the Orion program that's being run, looking at inclisiran, the way that invitations are done is by seeking information about patients from NHS Digital, and then we work out which parts of the country, which hospitals have the largest numbers of eligible patients, and then focus on the large centres where there are lots of patients to invite them into the study. And so collaboration with NHS Digital has been very important as a way of making the process of identifying patients very efficient. So I think that this process of trying to work with NHS Digital in the UK and other organisations elsewhere in the world who have large sort of collections of patients that can be followed using electronic health records is a key for doing this type of trial really efficiently and at low cost.
And going back to adaptable, when Giovanna and I wrote a comment, which is being published in the European Heart Journal, we noted that it was rather unusual and perhaps disturbing that no specific pathophysiologic hypothesis was put forward to justify testing a higher dose of aspirin versus a lower dose of aspirin. In other words, were they testing the hypothesis that aspirin had an effect on COX-2, which requires higher doses? Were they testing the hypothesis that aspirin was targeting a non-COX related mechanism of disease? There is no trace of any mechanistic reasoning either in the introduction or in the discussion of that paper. And we found that quite unusual. What do you think about it?
Well, I do agree that it would have been helpful to have a more clear statement of why the hypothesis was assembled as it was. But I think that it is a clinically relevant question, because in the US, as in Europe, both doses are used and nobody knows which dose is preferable. So I think it's possible to have a trial that is designed to settle a controversy without necessarily having a hypothesis that is mechanistic. However, if you're going to do that, then you have to be able to say why you chose 15%, because that's what they chose. They chose to be able to demonstrate that 325 was 15% or more better than 81. And certainly the evidence from our own work in the Anti-Thrombotic Trialists Collaboration, which we'll talk about maybe later on, doesn't really support the idea that might be differences that big between doses. Nevertheless, and we still don't know, because the way in which randomization or drop-ins occurred from the higher dose to the lower dose in adaptable really prevents us from drawing any clear conclusions from the study. I think the idea being that a 15% difference is not ruled out by the study because of that problem within the trial. One
way or the other, because actually 81 might be conceptually better than 325 by sparing vascular prostacyclin biosynthesis, which we know from the COXIB and NSAID experience does seem to make a contribution to.
That's possible, yes. Well, I'll wait for you to come and we can do a trial to see if we can discover that.
Okay, and on the other hand, if we turn to the ASCENT trial, as you know, the results of the ASCENT trial have informed the most recent European guidelines on the management of patients with diabetes in defining the role of aspirin in the primary prevention of serious vascular events. However, while ASCENT recruited people with diabetes without any threshold of estimated cardiovascular risk, the ESC treatment recommendations have restricted consideration of aspirin patients with diabetes mellitus at high, very high risk, who represented only a minority of the assigned population and for whom there was no evidence of a more favorable benefit risk profile than in the whole population. So is there an apparent disconnect here between evidence and treatment recommendation?
Well, I can only say what I think. As you know, the dilemma we have with using aspirin is that it causes bleeding. We know that it reduces the risk mainly of myocardial infarction in the context of primary prevention. It doesn't have much effect on stroke because it causes some hemorrhagic stroke and reduces the risk of ischemic stroke. So the two are balanced out. And the bleeding is chiefly gastrointestinal. And when you look at the maths, it turns out that in the populations that have been studied in primary prevention trials, in absolute terms, if you count an MI as the same as a bleed, then they roughly balance out. So many have said, well, what we need to do is we need to choose a higher risk primary prevention person, because then we'll have bigger benefits and then the bleeding won't be such a problem. But the problem is that when you do that, when you choose the slightly higher risk primary prevention patients, their risk of bleeding turns out to be higher too.
Sure.
And so you don't actually end up with a win. And this is true, I think, for diabetic patients or people with diabetes, just the same way as it is for other primary prevention contexts. So I don't believe that, I mean, we can talk about whether a bleed really is the same as an MI, and I think there is an argument that it isn't. But just purely on the counts, the risks of bleeding are similar in all types of primary prevention patients. However you try to push up the risk, you will push up the risk of bleeding at the same time.
And the other difference is that upper GI bleeding can be substantially prevented by rational use of PPI, as shown by your own work.
Yes.
With the meta-analysis of gastroprotectant agents.
Yes, absolutely agree. In fact, that's something we've discussed many times. And I think that there's a need for a reappraisal of how we use aspirin in primary prevention because of that observation, and also H. pylori eradication, if it was used more systematically, potentially could improve the benefit risk ratio for the use of aspirin in primary prevention. But that's work that we're slightly getting ahead of ourselves, but that's the work that we are currently doing because we're updating our work on aspirin for primary prevention with some of the new evidence that's come forth.
Okay, so that brings us to your second field, main field of activity, that is meta-analysis of individual randomized trials. So first of all, can you remind us what different types of meta-analysis can be conducted with the existing published data and with individual participant data that are not typically available in the public domain?
Yes, meta-analysis is the idea that, particularly, I confine my attention to trials where meta-analysis of randomized trials can provide useful information for guiding clinical practice on particular interventions. And I've been interested in this right from the very first day that I arrived in Oxford, because Richard Peto and Rory Collins had already established this methodology together with Salim Yousuf, who'd been a DPhil student in Oxford. And I really was immersed in this right from the very beginning. And the first thing I worked on was the fibrinolytic therapy trialist collaboration, where, if you remember, in those days, people used fibrinolytic therapy to open arteries after acute MI. Obviously, that technology has been replaced in many parts of the world now, but in those days, that was the question. And the question was, we had about 9 trials. What were the effects of fibrinolytic therapy in different contexts? But to step back a bit and answer your question, how does it differ to have individual data from the trials to just simply looking at the results of trials? And that can be answered by thinking about the evidence that there was before the large trials like ISIS-2 and GC came along. There were lots of small trials, and it was possible to just take the results from the publications and analyze those appropriately using standard meta-analytic methodology to learn that the most compatible estimate of effect of treatment was around about a 20%, from memory anyway, 20% reduction in mortality. So although it didn't prove anything, it sort of gave you an idea of what the likely effect size might be. And in the context now of some questions, it's possible to get useful information just by taking the published data for the overall effect of treatment. That's what you get. You get some idea about the overall effect of treatment, maybe on a number of different outcomes. But it doesn't tell you anything about the way in which the treatment works, how long it takes to have an effect, whether it is more effective in for particular outcomes, in particular vascular beds, say, or whether it has particular hazards in particular people, for example. That type of analysis really requires the effort that goes into bringing together all the individual patient data and then analyzing that in much more detail. And that was a project I first got involved with when I came to Oxford. And many will know that resulted in the idea that treatment should be given within the first 6 hours, but earlier the better. And then there was all the argument about the golden hour with, some of the trials going on, just looking at very, very rapid use of fibrinolytic therapy and so on. So it also suggested that there was an early hazard of giving thrombolytic therapy. And this was particularly in people, for example, with ST depression. And so we did get quite a lot of information about this particular type of treatment from having the individual data. And since then, I've been interested in using individual patient data meta-analysis to try and develop a mental map of how treatments work. And I do think of it like a map. If you have if you go to a new country and you have lots of small maps of different areas, then it doesn't, in strictly speaking, you could say, well, this is the country, but it doesn't tell you anything. What I want to know is, I want to get underneath the skin of a treatment and really understand why it is working in the way it does in different types of patients. And to give you an example of that, if you think about the work that we've done on the cholesterol treatment trialist collaboration, this is the work looking at all the statin trials, statin versus control and more intensive versus less intensive statin. This work has been enormously helpful in understanding how to use lipid-lowering therapy over the years. But I remember a time when the idea was put forward that you could perhaps do something for people with advanced heart failure or New York Heart Association 2/3 heart failure. And then the results of 2 trials in succession were negative. And we were able to understand, perhaps what we should have realized before we started, is that since we know by looking very carefully from the meta-analysis that statins don't prevent the types of death that occur in this type of patient, they really only prevent atherosclerotic types of death. They don't prevent sudden death, they don't prevent arrhythmic death, they don't prevent pump failure death. We could have perhaps predicted that there shouldn't really be much of an effect in heart failure patients. And so by understanding how treatments work in very great detail and the timing of their effects, one can start to understand why trials gave the results they gave and you can predict what might happen in the future. So to give another example from the CTT, the observation in Fourier, the trial of the PCSK9 inhibitor, was a bit disappointing, really. I mean, you expected a bigger reduction in coronary events in the early period than we saw because of the size of the LDL reduction. Mm-hmm. But actually, what we learned from the CTT was that when you lower LDL cholesterol with a statin, in the first year, there's only about half the effect that you eventually see in the second and third and fourth years. So if you have a short-term trial of LDL lowering, then it's dominated by that diminished effect in the first year. Mm-hmm. And the Fourier trial was only a couple of years mean duration, and that's why we should have realized that's what we would see if we had a short-term trial. So I think that, and the reason I love it so much, frankly, is because you learn so much about how something works. And I always, as a kid, I wanted to take things apart and find out how they work. Now I have this opportunity to find out how treatments work without taking anything apart and annoying my father.
Only numbers.
That's right. It really is like detective work. It takes forever to do, of course. I mean, several decades before we got to this stage. But in all seriousness, the amount of information we now have available about statins has had a major impact on human health. Is contributed substantially to improvements in human health. But we wouldn't have got there if we hadn't taken the trouble to assemble the evidence in a way that could be understood properly. I don't think of meta-analysis as being a kind of mechanical process of just sticking numbers into a computer. For me, it really is a detective work.
Mm-hmm. Now, typically, these kind of meta-analyses have been used to assess drug efficacy, but you have provided a very good example of how the same tool can be applied to assessing safety in the case of the whole class of non-steroidal anti-inflammatory drugs. Can you comment on this particular aspect of applying IPD meta-analysis?
Yes, the so-called CNT in meta-analysis was a very good example of something that turned out to be much more interesting than I ever thought it would be. If you cast your mind back a few years and to the time when everybody was very concerned about Vioxx, rofecoxib, and the hazards of Vioxx, and then there was quite a lot of hyperbole about the dangers of these drugs. I remember somebody saying that you were flying a jumbo jet into the Twin Towers every day. This kind of thing really it just called out for trying to get a slightly more measured approach to understanding the hazards. These are drugs that are extremely important to our patients. I remember being at the Health Canada hearings about non-steroidals, about COX-2 inhibitors, and patient after patient came up to the microphone to say, please don't take these drugs off the market. Please don't. I can't get out of bed in the morning unless I take one of these drugs. So for me, the challenge is trying to get good information about the benefits of the drugs and their hazards, and to be able to tell a patient, look, if this drug is something that you want to take, this is what you have to accept in terms of hazard. So that was the reason why it was important to me to get involved in this area. And what we were able to do, as you remember, you and I talked about this, was to take the trials of a COX-2 inhibitor versus a non-steroidal, a traditional non-steroidal, and look at those trials. And then we also took the trials looking at a COX-2 inhibitor versus placebo. And at first sight, you might think, well, why would you do a trial like that? These are painkillers. But it turned out that, very fortunately for us, these drugs have been studied in the context of chemoprevention and a few other conditions where it is possible to randomize patients to a placebo and a COX-2 inhibitor. So we had these two types of trials, and then we were able to compare the two results. So conceptually, if you know the difference between a COX-2 inhibitor and a non-steroidal, and the difference between a COX-2 inhibitor and a placebo, then by doing indirect comparisons, you can work out roughly the difference between a non-steroidal and a placebo. And there never have been any trials, long-term trials, of traditional and non-steroidals. But by doing this, we were able, as well as quantifying the risks of COXIBs, we were able to quantify the risks of traditional non-steroidals. And what we discovered was in line with other types of studies, but more reliable than the observational studies. We discovered that naproxen, which has a sort of aspirin-like type effect, you taught me this, so I'll say it, that the half-life of naproxen is long enough that you can get sustained inhibition of COX-1 during the treatment period. So you get a sort of aspirin-like effect, and that means that there's less of a hazard for naproxen in terms of its effects, adverse effects on MI, than we see for something like diclofenac, where the hazards are just as great as they are for high-dose COXIBs. And so so this is an example of where we had to organize the data in the right way before we even started the meta-analysis, because it was the indirect comparisons that actually led us to being able to quantify the risks, not only of COXIBs, which was what everybody wanted, but also indirectly of non-steroidals. So meta-analysis has many different uses, and we could go on because we've always done meta-analysis using very straightforward statistical methods. But when we came to look at another collaboration that I've been involved with, the Treatment Trialists Collaboration, where thrombolysis was being used to open cerebral arteries, there we had to actually use multivariate methods to understand the treatment effects, because we had such confounding that we absolutely had to have models. We couldn't do it in a simple way. So if anybody's mathematically inclined and starting out their careers, I can commend the idea of getting involved in meta-analysis because it is such fun.
Okay, thank you, Colin. Massimo, would you like to take over?
Well, I have to say that I enjoyed very much the enthusiasm of Colin in his work and in the applications, and just sticking to the individual participant data and meta-analysis. Very recently, the blood pressure lowering treatment trialist published an interesting IPD meta-analysis in a huge number of subjects in the Lancet, and the conclusion of this was pretty strong because it seems to affect immediately guidelines. They say the conclusion, the findings call for revisions of clinical guidelines recommendation globally. It suggests that the antihypertensive medications are better viewed as treatment options for prevention of cardiovascular disease, regardless of the individual blood pressure baseline level and the previous history of cardiovascular disease. And this is a revolution in the future blood pressure guidelines if they approach this way. But we commented this in our weekly journal Scan, and our conclusion was that this was very much reminiscent of what happened to cholesterol. And the conclusion of our comment was that the finding is reminiscent of the meta-analysis of the cholesterol treatment trialist, which shows more or less the same, that statin therapy can reduce 5-year incidence of coronary events and other MACE by about 1/5, largely respective of the initial cholesterol level and the risk profile. So, in other words, they emphasize the need to define high cholesterol or to define the floor level for cholesterol reduction. What is your opinion of this, parallel between these two analyses and, in general, the impact of this meta-analysis on guidelines?
Thank you, Massimo. Well, this is something that didn't particularly surprise me because if you think about let's just talk about the cholesterol treatment trials to start with, and so that put it in context. The evidence from the CTT flows from the evidence in epidemiological studies. So if you think about the observational studies, we know that for LDL cholesterol, each millimole increase in LDL cholesterol it results in around a doubling in the risk of coronary heart disease. And there's no lower limit that we know of at this point where that's not true. So there's this continuous relationship right through the range of studied LDL cholesterol. And we have the same phenomenon for blood pressure, incidentally, but I'll come to that in a moment. But if you know that epidemiology and you know the trials, which now show that it doesn't matter how long you go, lower LDL cholesterol is more beneficial. So lowering cholesterol is beneficial, however low you go. And then you choose, so we don't want to take very low risk people and lower their LDL from 1 to 1/2 millimoles. So what we want to do is take the very high risk people who are still at very high risk, despite having low LDL cholesterol. And we now have the drugs that can lower it still further. So that journey we traveled on over the years, first of all, we were focused on people with the highest cholesterols back in 2-3 decades ago, because they were the ones who were pitching up in FH clinics and, who were our main focus, and LDL cholesterol population-wide was much higher then anyway. But we've learned now about the epidemiology, and we've learned about how LDL cholesterol is a causal risk factor, and we can reverse it using various lipid-lowering drugs. Now switch the word lipid lowering to blood pressure lowering, and actually the structure of the problem is very similar. We have log linear relationships between blood pressure, systolic or diastolic, and the risk of vascular disease, whether that's atherosclerotic or structural. And it's no surprise that we see something very similar because it's a similar structure. The difference, though, is important to note, and that is that you cannot simply keep going lower and lower blood pressure with your drugs. At some point, the patient will complain and they won't be able to tolerate it. And so there's a similarity, but there are differences. For clinical practice, I don't know whether this will be something that will result in changes in the future guidelines, but they will have to be nuanced because it isn't exactly the same.
One final question. Thank you very much. One final question is, I don't want to take you much longer. This is a very tough question, but I hope you can synthesize. Can you comment or share with us the key functional elements and rules underlying the ESC CPG guidelines committee that you are chairing? So in other words, what is the strength of evidence of different approaches like randomized trials or meta-analysis or other approaches, and other rules like conflict of interest and so on? Yeah, I know that this is a very integrated and complicated question, but maybe you can just share with us the key elements of
Sure. Well, I took over from Professor Vineka last year, and there were some clear things that I wanted to try to advance forward if we could. And they really have to do with trying to ensure that the ESC has the best guidelines in the world for use of cardiologists and associated healthcare professionals who are looking after cardiological patients. At the moment, everybody likes the, certainly in the profession, there's a feeling that the cardiovascular guidelines that we produce are readable. They're in a format that can be easily used, and there's a lot that we don't want to change about them. But I think there are some things that are in need of change. You mentioned a few of them, and I'll just go through some of the things that I'd like to try to achieve or set on in progress whilst I'm chair. The first one of these is the methodology, many people have said that ESC guidelines are not reliable because we don't do systematic reviews for every question. And I have to say that I don't really agree with this, because I think that for many questions, we have large trials or large studies which are on their own informative, and doing a systematic review simply assembles the bits that we know about, which are decisive, together with the bits we don't know about because we haven't done the literature searches, which are probably going to be indecisive. So I think we have to be very careful about how we marshal our resources to incorporate systematic reviews. But I think there are some cases where we should be taking a much more systematic approach to assessing the evidence that we use in our guidelines. And so as part of the effort to consider that point, we are experimenting with using the so-called grade methodology, which is used in many guidelines, for one of the ongoing guidelines at the moment, pulmonary hypertension. And we'll see how that goes. We'll try and learn from it. But ultimately, I think that we want to have at least some of our guidelines using systematic approaches to the evidence to tackle particular questions that are clinically very important. And I'm thinking particularly where you have parts of a guideline where, say, a new drug or new drug class has come forward. SGLT2 inhibitors would be a very good example. Something that's happened very recently, lots of evidence coming forward and the need to review it before you write a guideline. I think that typically for guidelines, we might have 1 or 2 or 3 or 4 maybe questions where we would want to have systematic reviews planned at the time of the guideline. And for the remainder, we can probably not use systematic reviews, although we do want to have a systematic approach to gathering evidence. So that's the first thing. The second thing I really dislike and I want to change is the way that we categorize the levels of evidence, because I think that the system that we have at the moment is not optimal at all. And we have situations, for example, where an observational study, a large observational study is equated with quality of evidence that we would get from a large randomized trial. And this is simply not appropriate. Observational studies can never be as dependable unless we're looking at very large treatment effects as randomized trials. And so I want to review the way we do that. The ideal way to do that, I think, would be to adopt the grade approach, which is much better in my view, but this is work in progress. So we're still thinking about how we do that exactly. The second area that I think needs attention is the way in which we have an assessment of conflicts of interest. And this feeds into another area which I think needs attention, and that is how we select people for service on task forces. There's a tendency to, because the opinion leaders are opinion leaders, and we often go to them because we depend on their judgment, I think there are many younger people who would benefit from being involved in task forces and contributing. And so my aim is to try to widen recruitment for task forces in future guidelines, and that's something that the committee is absolutely behind. We also have movements within the ESC to limit financial interests to a great extent than we have done previously, for not just for the chairs of task forces, but also for task force members. So from those guidelines reporting from 2024 onwards, it will no longer be possible to serve on a task force if you have financial income from industry of more than 10,000 euros a year. And this is a big change, but it's something that I think that the public expect us to incorporate into our approach. Those are the main things that we're focused on at the moment: methodology and getting conflicts more transparent. The other way in which conflicts, I think, can be managed is to do something about at least have a slightly more rigorous voting process, so that people who have very specific conflicts within a task force are prevented from voting for particular questions when they're assembling the guidelines. This is not something that we've incorporated yet, but we're currently thinking about this and trying to come forward with proposals. So I think that there has to be a clear, the public have to be able to trust what we do. And I think at the moment, there are some things that we can improve, whilst not wishing to break the clearly excellent work that is done or has been done, and produce guidelines that cardiologists like. I mean, they're downloaded huge numbers at times. There's clearly a very valuable resource, but I think we can refine them and improve them using the types of approaches that I've mentioned.
Thank
Colin,
you very much.
your last question was very stimulating. I'd like to ask one more question to Colin. Colin, you are very, very creative, and guidelines are excellent documents, but I'm sure that you will improve them. Also, what is excellent can be further improved. And really, with your experience, you are the right person to improve guidelines even further. One last comment about guidelines is the length. They tend to be very long and sometimes difficult to read. What are you planning for, perhaps shortening them or improve readability, or come up with an executive summary? What do you have in your pipelines in this regard?
There's nothing specific at the moment, Filippo, about length. We, as you know from your involvement as the new editor, we're pretty strict with the task force chairs about length. We try to work with the task force chairs during the creation of guidelines to help them to shorten where this is possible without loss of clarity. And as I have to say, that certainly the task force chairs that I've worked with are really focused on trying to make documents that are readable and don't get bloated. But you know, part of the trouble is that these cardiologists have a habit of creating evidence, and so we have more and more that we have to cover each iteration. It's not a problem I think that we need to worry about, but we are not only faced with the problem of expanding evidence, but also expanding indications. You know, the areas of medicine that get covered tend to expand. One thing that we're hoping to do for the future is to potentially combine some of the titles that we've traditionally looked at. So in the present round, for example, we are combining a guideline that used to be split into non-STEMI and STEMI ACS. We are now looking just at ACS. So there's going to be one guideline on ACS for 2023. And that was a way of trying to make guidelines more practical. So if a patient who comes through the door doesn't have NSTEMI written on their forehead, they have to go through a process before we know what management pathway they go down. So we're trying to make them useful documents that can be used on the ward.
Thank you, Colin.
Okay, it's 6 o'clock exactly. So I'd like to thank first Filippo Crea for coming with the idea of organizing these interesting dialogues. My team, Giovanna Liuzzo and Massimo Volpe, with whom we thought about it, choose the topic and the ideal person to talk to. Thank very much Colin Baigent for very interesting and stimulating interaction, and all of our readers who have been following this dialogue either through the Zoom call or are watching us on YouTube in streaming. So thank you all, and have a nice evening.
Carlo, one last thing from me. Well, of course, I join you in thanking Colin for his participation to the dialogue. But let me thank you, Giovanna and Massimo. You are the editors of the weekly journal Scan, and I wish to thank you for your extraordinary contribution to the journal. This is a new feature of the journal, which is becoming increasingly more popular. It's a new window we have opened on seminal papers published in important general medicine journals like Lancet, New England, and JAMA, accompanied by your editorial, short but interesting editorial comment. This feature is becoming more and more popular, and as editor-in-chief of the journal, I wish to thank you for your contribution to the journal. And one last thing is next dialogue. I remind you that, as usual, the dialogue will be on the last Thursday of the month. Next dialogue will be on July 29th at 17 Central European Time, as usual. Thank you again. Thank you to all of you. Bye-bye.
Thank you all.